Recording. Today we're going to begin talking about DNA. We'll talk a little bit about, there is a study guide for the genetics test that's posted. And this genetics test coming up as well as the last material that we'll talk about with DNA. As I mentioned a week or so ago, it's mostly problem-based. So there will be some multiple choice and true false and things just like we've had on earlier tests. But there will be a lot of problems where you'll have to work a genetic problem. So it's really crucial to work on those Mendelian genetics problems and non-Mendelian genetics problems, the last two assignments, if you haven't got started yet, because that will be probably half or more than half of the points on the test on Monday. So you definitely want to know how to do those. And keep in mind for homework, if you have questions, you can always email me and ask questions, or there's a couple people that we've had separate Zoom meetings and talked about things. So on the test, I won't necessarily, go through step by step how to work a problem with you, but on the homework, I can definitely help you if you have any questions. So keep that in mind. The other thing is, remember what I've said on several occasions, if you would like to see your homework with the corrections, once you turn it in, up by where you type in your name, tell me that, please send me my corrected homework back or whatever. And I'll send you an attachment back and you can look at that. And we can talk about it if you don't understand why you missed something or how you should do it correctly. So a couple things to keep in mind. So next week we'll have a test over genetics on Monday. You can probably tell Monday is our typical test day. Now next week is the last week of class. So the problem is we don't have a Monday after that. So besides the test on Monday, we will have a test on Thursday on DNA, which is the topic that we're starting today. So we're going to talk about DNA some today, some Monday after the test, and then Tuesday and Wednesday. And again, we will have some problems. So there's an assignment up that is due on Tuesday. And on Monday, we'll have one that's due on Wednesday. So it's really crucial to work these practice problems so that you're not trying that type of problem for the first time when we get to the exam. Any questions? Okay, well, today we're going to focus on two basic topics. We're going to go back and look at the structure of a DNA nucleotide to begin with, and then the entire DNA molecule. And we talked about this back when we discussed chemistry. We're not really going to go into much more detail now than we did then, maybe a little bit. But now that we've looked at how characteristics are passed down from parents to their children, the next topic that we're going to focus on is what role does the DNA play in this passing down of characteristics? What's happening on a chemical level? And so we're going to look at two processes that we've mentioned before. When we talked about the cell cycle, we talked about replication, cells making the sister chromatids, the identical versions of their DNA. And so we're going to look at chemically what happens during replication. And the other thing that we'll look at, we won't get to today, is we've talked on the level of the parts of the cell, how a cell makes proteins. We're going to talk about on a chemical level, how a cell makes proteins. So what actually happens during protein synthesis. And those will really be our two main topics. We'll look a little bit at how cells control when they make proteins, but basic focus is going to be DNA, how it's replicated, and then protein synthesis. Now, one of the things to keep in mind, we'll talk about something today called complementary base pairing. And we're going to ultimately talk about three different sets of rules for complementary base pairing. And these rules are really crucial. If you know these rules and if you know when to use them, you can work probably 90% of the problems that we're going to work. So you want to really focus on these rules when you're doing some of your study. So here's a DNA nucleotide. And remember, nucleotides have three parts. They have a molecule of deoxyribose sugar. That's where the D in DNA comes from. They have a phosphate group. And then they have a nitrogenous base, a base that contains nitrogen, which basically means either an amine or an amino group, either a nitrogen with one hydrogen or a nitrogen with two hydrogens. Now, there are four different nitrogenous bases. There's adenine, cytosine, guanine, and thymine. This particular example shows thymine, but it would look pretty much the same. Guanine, cytosine, adenine are a little bit different, but they'd be this green part over here on the side. So when we look at a molecule, one of the things we'll see is that we're almost always just going to abbreviate the nitrogenous bases. We're going to write A, C, G, or T. We'll hardly ever write the name out. But the other thing that we want to keep in mind is that it's really these bases that are the crucial part. A typical DNA molecule in your body is made up of about 150 million nucleotides. That's an average chromosome. Some have more, some have less. But when we think about those 150 million nucleotides, the deoxyribose sugar group is the same on every one of those. The phosphate group is the same on every one of those. The only difference is whether you have A, C, G, or T. So when we think about two people and we think about their genetic differences, if we reduce it down to kind of the most basic chemical level, the reason that you have blue eyes and somebody else has brown eyes is because of the order of the A, C, Gs, and Ts. So we're going to be focusing on the order of these letters for a lot of what we do. So this is one nucleotide, but I just said that a molecule of DNA on average has about 150 million nucleotides. So let's look at how these are joined together. And the way that they're joined together is we have the deoxyribose sugar, and it has 5 carbons. So it's always represented as some sort of pentagram. And we have the phosphate group, which is purple here. And notice that the deoxyribose sugar and its phosphate, the phosphate is connected to the next deoxyribose sugar. So we have a kind of a chain of deoxyribose sugars and phosphate groups. And then sticking off to the side are the nitrogenous bases, the A, Cs, Gs, and Ts. Notice that they're not necessarily in alphabetical order in real life like they're showing here. Now, if you think about this, this kind of looks like half of a ladder. You know, the side of the ladder, what's called the riser, is the sugars and the phosphate groups. And then we have half of a step, which is this nitrogenous base sticking out in the middle. This is important because when we look at DNA, it turns out that DNA is actually, even though this would be one molecule of DNA, when we look at the DNA in your nucleus, we don't see DNA in this form. When we look on a chromosome, whether it's your nucleus or the nucleus of an oak tree, or whether we look in a bacteria cell, which doesn't even have a nucleus, remember, it just has a nucleoid with one single chromosome. When we look at a DNA molecule, it's actually 2 molecules of DNA that are bonded together and twisted around each other. So let's look at how this is twisted around. So remember, we said that our molecule of DNA is kind of like half a ladder. So imagine the other half of the ladder over here. And in the other half of the ladder, the nucleotide, the nitrogenous base, sticks out into the middle again. And we have something that we've only seen in one other molecule that we've talked about so far this semester. Remember back when we discussed water, we said one of the things that makes water a unique molecule and gives it properties that are very important for living organisms is that in water, we have hydrogen bonds. In DNA, we have hydrogen bonds. So when we have another chain of nucleotides over here, there are going to be hydrogen bonds connecting the nitrogenous bases on one chain to the nitrogenous bases on the other chain. So this is how we hold DNA together. And hydrogen bonds are very useful because if you remember when we talked about water, hydrogen bonds are very weak. And this is good because, as we're going to see, if we want to make a protein or if we want to replicate our DNA, we're going to have to pull our two strands of DNA apart. So it's good that these bonds are weak. But at the same time, think about what I just said. There's 150 million nucleotides in one of these molecules. And every nitrogenous base has either 2 or 3 hydrogen bonds to the nitrogenous base on the other strand. So that means there's 300 to 450 million hydrogen bonds. So even though individually these hydrogen bonds are very weak, when you get that many, they hold the DNA molecule together pretty firmly. So it's held together firmly, yet at the same time, it's easy to separate the two strands. So this is really an important property that these hydrogen bonds give the DNA. So let's imagine we have another strand here, and let's take kind of a glance at what our ladder looks like. Now, it doesn't look like an ordinary ladder. Probably a better way to think of it, imagine you have a ladder and you plant the two sides of the ladder into the ground and then you twist it so that you have a kind of a spiral staircase. That's what we have with the DNA. Now, because we have two molecules of DNA twisting around each other, this is called a double helix. And if we look at our double helix, the kind of bluish gray along the side, this is the alternating sugars and phosphates, sugars and phosphates, sugars and phosphates. And usually we just kind of don't pay that much attention to them because, as I said, they're always the same. So we want to focus on kind of the rungs or the steps on this ladder. Now, notice if you look at this ladder and you focus on those rungs, there are 4 different colors. There are some green bars that represent adenine. There are some black bars that represent guanine. There are some blue bars that represent cytosine. And there are some red bars that represent thymine. Now, notice if you look carefully, these pairs of nitrogenous bases are not just arranged any old way. If you look, red and green always make a step, or blue and black. You know, if you look down this representation of DNA, we never see blue bonded to red or blue bonded to green. So when we have a cytosine on one strand, it's always hydrogen bonded to a guanine on the other strand. Those two form a unit. If we have an adenine on one strand, it's always bonded to a thymine on the other strand. And the fact that A always pairs with T and C always pairs with G, this is called complementary base pairing. Now, this is not complementary with an I, this is complementary with an E. And complementary with an E refers to things that complete each other. Back in geometry, I think complementary angles, if I remember right, added up to 90 degrees. In this case, A can only pair with T, C can only pair with G. And this was actually discovered in 1953 by 2 scientists named Watson and Crick. And Watson is actually from the Chicago area. He was kind of a child genius. He graduated from the University of Chicago when he was like 18 or something, or 20. Crick was in London and Watson went to work with him. And they figured out how complementary base pairing worked. Now, the way they figured it out was they basically had a set of big metal tinker toys. And they were trying to fit the pieces together to see how they could explain what they observed when you took photographs with X-rays of the DNA. And one of the things that you observed if you took X-ray photographs of the DNA was the molecule was basically the whole same width throughout its whole length. So, you know, it's hard to tell representing a three-dimensional molecule here, but even though it twists around, the molecule was basically the same width across everywhere. And this didn't make much sense because if you look at adenine and you look at cytosine and you look at guanine and you look at thymine, they're all different sizes. So how is it possible that the molecule is always the same width if each of these four nucleotides is different? Well, it turned out when they were playing with their big metal tinker toys, what they discovered was even though all four of the nitrogenous bases are different sizes, if you look at an A paired with a T, an A paired with a T, and a C paired with a G are the same size. So the individual nucleotides aren't the same size, but the pairs are the same size. And this was really kind of the aha moment that helped Watson and Crick discover the importance of complementary base pairing. Now, from our point of view, when we think about this, we can look at some small sections of a molecule of DNA. And here we see a thymine and on the left side and an adenine on the right. And in the middle, we have two dotted lines. These are hydrogen bonds that form between the thymine and adenine. We have below that a guanine on the left and a cytosine on the right. And we have three dotted lines. They form three hydrogen bonds when they connect. Now, the two strands of DNA are described as being anti-parallel. And again, back in one of those math classes, probably geometry, you learned about parallel lines that basically go in the same direction. Anti-parallel refers to things that go in an opposite direction. So let's go back for a moment. So notice when we look at one of our molecules of DNA, we have something that's called the 5' end. And the 5' end is where the end of the molecule is a phosphate. And then we have the 3' end, and the 3' end, the 5' end, by the way, is named because deoxyribose has 5 carbons and the phosphate is bonded to the 5th carbon. The 3' end is just this carbon atom sticking out down here. So when we look at this, we always think of DNA or RNA when we talk about it in a little while. We always think about DNA as starting at the 5' end and going to the 3' end. And just like when we see an A, we know it's going to be paired up with a T if we would write the other strand over here. If we have a C, we know it's going to be paired up with a G over here. The 5' end of one of the strands is always paired up with the 3' end of the other strand. So remember, we said we're thinking of starting from the 5' end, from the phosphate group. So if we would draw in our molecule on the right here, on that, the 5' end, the phosphate group is on the bottom. So because of this, we say that our two strands of DNA are anti-parallel. Notice they show an arrow going from the 5' end towards the 3' end. And all these little blue boxes, notice again, all these little blue boxes, these represent the deoxyribose sugars connected to the phosphates. Since they're always the same, you know, keep in mind what I said, we're going to focus on just the letters that represent the nitrogenous bases. A lot of times we don't even draw the sugars and the phosphates. We want to keep in the back of our mind that they're there, but, you know. And again, notice if we look at this time A is represented in red, T in yellow. And if there's an A on one side, there's always a T on the other side. Notice the A can be on the left and the T on the right, or vice versa. You know, if we have a C on one strand, the blue, and a G on the other strand, green, they're always paired with each other. So when we look at an actual chromosome in your body, technically it consists of two molecules of DNA. Now, the two molecules of DNA are complementary, if we would look at all these nitrogenous bases, and they're held together by these hydrogen bonds, and they kind of twist around each other like a spiral staircase. So we want to keep this in mind when we're thinking about our DNA. Now, when Watson and Crick discovered complementary base pairing, the first thing they did was they said, oh, this explains how a cell can replicate its DNA. This explains what happens during replication. Because replication was a mystery to biologists. They didn't know exactly how it happened, but they knew it was very, very accurate. During replication, one way to think about this, if we would just focus on, say, the left strand of DNA, if you would look at all the DNA in the human nucleus, there's about 3 billion nucleotide pairs. So 3 billion letters, A, C, G, and T, and then they're paired with, you know, whatever their complement is. So 3 billion letters. When your cells are getting ready to divide by mitosis or meiosis, remember that they replicate their DNA during the S phase. And S takes different lengths of time in different cells, but typically it takes a few hours. So in a few hours, the cell can accurately replicate 3 billion nucleotides. This would be like me giving you a book that was 1,000 pages or so, and actually 3 billion letters would be about 20 copies of that book. And it would be like you being able to type that entire book over accurately in a couple of hours and only make a mistake about once every billion letters or so. So replication is very accurate and very fast. But as soon as Watson and Crick discovered complementary base pairing, they immediately said, oh, we know how replication has to happen. We know why it can be so fast. And even though it's fast, why it can be accurate. Because, you know, usually if you rush things, you make more mistakes. In this case, Watson and Crick began looking at replication. So let's look at replication. Let's tilt our DNA molecule on its side. And let's think about all the different enzymes that are involved in replication. So in this particular diagram, our DNA is represented by the two gray lines. Notice that over here on the far left, the two gray lines are together. These are our two strands of DNA. Over on the right, they're separated. One is up here on the top, one is down lower on the bottom. Now, when we look at replication, remember, the only time it ever happens is during that S stage of interphase, either before mitosis or before meiosis, depending on the cell. But otherwise, replication doesn't happen. So think about what we've discussed so far. To replicate our DNA, what we're going to have to do is we're going to have to basically end up with two identical versions of the chromosome that we start out with. So let's look at the different enzymes that are involved. And since replication started on the right, really in a sense, what happened at the beginning is this enzyme over here, because as replication starts, new enzymes come in. So the first enzyme that has an effect is an enzyme called topoisomerase. And remember what we said all semester, things that end in ASE, that's a hint that this is an enzyme. And what topoisomerase does is basically loosens up the connections between the nitrogenous bases on the two strands. It starts to kind of pry them apart. Now, it doesn't get very far. The enzyme that actually completes this job is this little blue enzyme over here something called helicase. And helicase unwinds that double helix. So over on the left side, we have that spiral staircase type pattern. On the right side, we've started to unwind it into two separate strands. Now, remember what I said, that when we think of DNA or RNA, we're always going to think of going from the 5' end to the 3' end. So we said the two strands are different in the sense that the 5' end on one matches up with the 3' end on the other. So on the bottom strand, the 3' end is over towards the left and the 5' end is to the top. And we have an enzyme called DNA polymerase that comes in. And it's actually DNA polymerase 3. There are several different types of DNA polymerase. And DNA polymerase basically starts over here at the first nucleotide. And let's say the first nucleotide is a C. It will add a nucleotide with a G. And then the second nucleotide might be a T, so it adds a nucleotide with an A. Now, where does it get these nucleotides that it's adding? Remember when we talked about the nucleus earlier in the semester, we said that there are kind of all sorts of chemicals in the nucleoplasm. We said the nucleoplasm was like the cytoplasm, and when we talked about the cytoplasm, we said it's like a warehouse. So basically, there are spare nucleotides in the nucleus, in the nucleoplasm, and this DNA polymerase has the ability to grab the right nucleotide and snap it into position. Now, how can it grab the right nucleotide? This is the importance of complementary base pairing. It grabs the right nucleotide because what? Because if there's an A down here, the only thing that will fit in next to that A is a T. If there's a G down here, the only thing that will fit next to it is a C. So this is why when Watson and Crick discovered complementary base pairing, they said, now we see how replication can be fast and accurate. Because really, from a chemical standpoint, it's hard for the cell to make a mistake. Mistakes do get made occasionally, but they're very, very rare. So here, the 5' end of our new DNA, this peach-colored strand, is on the right, because we always start at the 5' end. Over here, it's a little bit hard to see, but there is an arrow tip, so we can see that we're moving to the left. And what we see is that we're going from our 5' end down here towards our 3' end. Now notice, we're going from the 5' end of the new DNA to the 3' end of the new DNA. We're actually starting at the 3' end of the old DNA because of the fact that things are complementary here. So we want to keep in mind that's going on. Now, until we get these new nucleotides attached, the molecule isn't very stable. So after helicase splits things up, we have some molecules called SSB, single strand binding proteins, that come in and attach to the strand of DNA and basically stabilize it till the DNA polymerase can come along and put on the complementary base. So these single strand binding proteins are kind of like a temporary placeholder. They're going to keep the DNA happy until we can add a new daughter strand. And again, the new strands are going to be called daughter strands. We don't have any son strands. Now, since replication overall is moving from the right to the left, and since replication here is moving from the right to the left, this is called the leading strand. The other strand is a little bit trickier, because notice on the other strand, the 5' end is all the way over here on the right. Well, remember, we said we're always going to think about the new DNA or RNA when we get there. We're always going to think about the new DNA being added from its 5' end. But if this is the 5' end of the parental DNA, the gray DNA, then this is the 3' end. So what happens is, instead of going in the same direction, the cell has to jump back a little bit. And it has to jump back. And at some point here, the cell is going to start making a piece of DNA represented by this green and peach. And it's going to start making it at a 5' location. And then it's going to go to the right, opposite to the direction that replication is going. So this is called the lagging strand, because if you lag, you're kind of behind. And this is a little more complicated. We have to have actually 3 enzymes that work here. We have to have something called RNA primase. And RNA primase puts down this little green section, which is actually a section, it's kind of like the start for the DNA when we have to jump. Then DNA polymerase comes in and does the work. And DNA polymerase does the same thing up here as it did down here. And then finally, we have an enzyme called DNA ligase. And the problem is, if we look at this leading strand, see how it shows these gaps between these little pieces of DNA. These little pieces of DNA aren't actually bonded together on the lagging strand. So DNA ligase comes together and it attaches this strand here to this strand. And then it'll move down here and attach this strand to this strand. So our parental strands both get replicated, but they get replicated kind of in different directions, if you want to think of it that way. Now, let's look at another representation of this. So here's one where we have the 3' and 5' labeled. I didn't want to have everything in one diagram. It's enough to cope with just to have some of it. So the 2 blue strands are our parental strands. One's kind of a sky blue, one's more a navy blue. So here's the leading strand being made from its 5' end toward the 3' end. On the lagging strand, that RNA primase part, that's the red. So notice if we look at the light green up here, its 3' end is to the right. So that means its 5' end is this red. So the cell kind of backtracks a little bit, starts out and makes some DNA, then it jumps again. And so it makes all these little pieces. And these little pieces are named after the scientist who discovered them, a Japanese scientist. So these are called Okazaki fragments. And this is what that ligase attaches into one long continuous strand of DNA. So let's think how we can represent replication. So here's a strand of DNA, ACGT. Well, first of all, notice if we have one strand of DNA, if I tell you what one complementary strand is, you can figure out the other strand. Because if I have an A on the left strand, I've got to have it paired with a T on the right. If I have a C, it's got to pair with a G. G has to pair with C. T has to pair with A. So these are our 2 parental strands. Now, keep in mind what we're seeing here, where the 2 blue parental strands separate, or back here where the 2 kind of more grayish parental strands separate. The 2 parental strands get separated. So that's represented by our black type. And then we have some of those free nucleotides that are just kind of floating around the nucleoplasm that come in and get attached. Now, notice that, think about these nucleotides, the daughter strand that comes in and attaches to the parental strand on the left. Notice that these red nucleotides have exactly the same order as the parental strand that's separated. Why? Because of complementary base pairing. So the red strands represent the daughter strands, the black are the parental. And these two pieces of DNA actually represent, when we go back to mitosis or meiosis, those sister chromatids that we saw at the beginning of either prophase in mitosis or prophase 1 in meiosis. And you might remember when we talked about replication, when we talked about the S stage of mitosis, one of the things that I said is we don't like to say that the DNA is copied, because if we think about the DNA being copied, most of us think of a copy machine. You put a piece of paper in, you make a copy. When you get done, you have your original paper and you have a copy. If we look at the DNA after replication, the chromosomes in the two sister chromatids both have what? They both have half of the parental DNA and they both have half daughter DNA. So we don't really have an original and a new copy. We have two chromosomes that both have half of the original. So when we talk about this, replication is sometimes said to be semi-conservative. If you conserve something, you save it, and semi means half. So in replication, you save half of the DNA for each sister chromatid. But this is why we don't like to say that the DNA is copied, because it's not copied. It's not the right term. We use replication, but duplication is still better than copy. So we don't want to say it's replicated. So now, even though replication is very accurate, there are occasional mistakes. And when there are mistakes in replication, these are called mutations. So let's look at some of the different types of mutations. And there are different ways to think about mutations. Just like you could divide people up into different categories by height, by shoe size, by where they live, whatever. You can think of mutations as being in different categories depending on what you're interested in. So one way to think about mutations is to divide them up by how much DNA they affect. So something called a point mutation just affects one nucleotide. And most of these are pretty obvious. This one is spelled wrong. This is a substitution mutation. So in a substitution mutation, we have something like AAA if we look at our DNA. Now notice, this is a good example of something I mentioned earlier. Notice we're not bothering to write down the sugar or the phosphate because that's always the same. So we have AAA and that becomes AGA. So what happens? Well, if we look at our middle nucleotide, we started out with an A and we substituted a G. So this would be an example of a substitution mutation. We can also have something called a deletion mutation. Now, deletion mutations were named a long time before we all had computers and used word processors, but a deletion mutation is basically what a deletion is on a word processor. We have AAA, and after that, one of the A's gets left out. It's deleted. And we also have insertion mutations. And after an insertion mutation, we have something like AAA, and we get an extra nucleotide stuck in. So it's AATA. So these are mutations that only affect one nucleotide. A chromosomal mutation affects a bigger part of the DNA, so at least two nucleotides. Now, you can have an insertion or deletion. That's a chromosomal mutation because you could have something like ACGG, and then after that you have ACG, and then you have CTG. So you actually inserted two letters in the middle, or you could have cut two letters out. And it could be more than 2. I just used 2 as an example. You have a duplication when 1 or more nucleotides gets copied extra times. So you have something like ACGG, and then you have A, and instead of 1 C, you have 3 Cs. So that would be a duplication. And it doesn't have to be just a single nucleotide. It could be several nucleotides. So there's a disease in people called Huntington's chorea. And in Huntington's chorea, this occurs in a gene where normally in this gene, people have somewhere between 10 and 35 repeats of the 3 letters, the 3 nucleotides, CAG, CAG, CAG. So most people have somewhere between 10 and 35 copies of this. Well, when the cell is replicating the DNA, you know, if you've ever had to make a stack of 35 sheets of paper or something, sometimes what happens is, you know, you get distracted, you lose track, and what do? Yeah, maybe you toss an extra sheet of paper on there just to make sure you got at least 35. So in the cell, when the cell replicates the DNA, when we have a lot of repeats like this, sometimes the cell, you might start out with 20 and the cell puts in 24. Does a couple extra just in case it missed any. The problem is, once you get more than about 35 repeats, you get a disease called Huntington's disease. And people who have Huntington's disease can have anywhere from 36 repeats of these 3 letters to about 120 repeats of these 3 letters. And in fact, we can actually do a genetics test, and when we do a genetics test, we can find out exactly how many repeats of these 3 letters you have. And we can give you some pretty grim news about Huntington's disease, because the more repeats you have, the younger you'll be when you get Huntington's disease. And the bad news is Huntington's disease is really a devastating disease. In Huntington's disease, people's brain loses the ability to communicate with their muscles. So a lot of times they kind of walk like a zombie or Frankenstein. Their joints are very stiff. They can't bend their joints. And a lot of times people with Huntington's disease ultimately die because the muscles that allow them to breathe lose contact with the brain. So a lot of times they just die in their sleep. But the problem is, up until that time, it can be very painful when you lose the ability to move. So, you know, we could get a genetics test. We could tell you how many copies of the repeats you have. And then we can tell you, yeah, plus or minus a year, you're going to get Huntington's disease when you're 38. And we can be pretty accurate. And part of the reason that that's a very cruel diagnosis is right now we don't have a medical treatment for Huntington's disease. So it's kind of like a death sentence. We don't know how long it will take to kill you, but we can tell you when you're going to start showing symptoms. Now, we can also have something called an inversion mutation. When you invert things, you turn them upside down. So basically, we flip the order. So we have something like ACGT, and it becomes TGCA. Remember how we talked about the DNA getting tangled? So sometimes when the DNA gets tangled, it's kind of in a loop. And while it's in the loop, things are kind of going backwards. So when those enzymes that untangle things come along, sometimes they untangle the loop, the snarl, and then they sew it back into the DNA, but they sew it back in backwards order. Now, we can also think of DNA as terms of how the mutations actually affect the amino acids in the proteins. You can have something called a silent mutation. Now, mutations always change the DNA. So a silent mutation changes the DNA, but it doesn't affect the amino acids. You still get the exact same amino acids in the protein. So a silent mutation doesn't really cause any type of genetic disease or anything. You can have something called a missense mutation. And in a missense mutation, you get a different amino acid. You should have an amino acid called leucine. Maybe you get glycine instead. And you can also have something called a nonsense mutation. And in a nonsense mutation, instead of getting an amino acid, you get something called a stop codon. Think of it like a stop sign. Remember, ribosomes actually make the proteins. A stop codon tells the ribosome, no more amino acids. This protein is done. Some mutations are a little bit harder to put into categories. There's something called a translocation. And in a translocation, sections of the DNA that are on one chromosome, like chromosome number 1 maybe, get moved to chromosome number 2. And there's something called a frame shift mutation. And we can get frame shift mutations from either insertions or deletions. And frame shift mutations are usually serious because they change a lot of amino acids. So later when we talk about making proteins, we'll talk about this in more detail. But when the ribosome actually reads the messenger RNA, and the messenger RNA came from the DNA, the ribosome reads 3 nucleotides at a time. So if we had some DNA and it was AAA, and then CCC, and then GGG, and then TTT, the ribosome is going to read AAA and put the amino acid that's the instructions for. And then the ribosome is going to find the amino acid that CCC is the instructions for. And then the amino acid that GGG is the instructions for. And finally, the amino acid that TTT is the instructions for. But if you have an insertion, suppose we put an extra A in here. Now when the ribosome is reading 3 nucleotides at a time, it reads AAA. And since we put a 4th A in here, now the next 3 nucleotides are ACC. And the next 3 are CGG. And the next 3 are GTT. And then that T that was on the TTT is on the next one. So notice we could potentially change a lot of amino acids. So for some of these mutations, like frame shift mutations, we hardly ever get a protein that works. So if this protein is crucial for how your cells function, a mutation like this could be fatal. So I keep saying that replication is accurate. So how accurate? Well, studies suggest that there's a mistake about 1 in every 100,000 base pairs. However, you have proofreading enzymes. And before replication is done, they go back and they correct most of the mistakes. So at the end, there's about 1 error in every 10 billion base pairs. So hardly ever, really. So remember what we said at the beginning. Complementary base pairing is going to tell us how cells accurately and quickly replicate their DNA. The next question we need to address is, how does DNA, where you've only got 4 variations on the nucleotides, do they have A, do they have C, do they have G or T? How can that be the instructions for thousands of different proteins? Well, we'll get to that tomorrow, Monday. But what we want to think about before that is how we can use this information about complementary base pairing to get some information about DNA when we're doing a DNA analysis. So suppose we have a piece of DNA and we know one of the strands. What can we figure out? Well, because we know that A always pairs with T and G always pairs with C, if we know one strand, we can write the other strand. And not only that, because A and T pair together and C and G pair together, we can use something called Chargaff's rule. Chargaff was a biochemist in the early part of the 1900s. And when he started analyzing cells, he found out that the amount of A was always almost exactly equal to the amount of T. And the amount of C was almost exactly equal to the amount of G. Now, at that time, Watson and Crick hadn't discovered complementary base pairing. So Chargaff didn't know why this was the case. But now with complementary base pairing, we know why this occurs. So suppose you analyze some DNA and you find out that it's 21% G. Okay, if this DNA that I'm analyzing is 21% G, what do I know? 21% C. I know it's 21% G. And not only that, but if you think about it, you know, all our DNA here is equal to 100%. Okay, if our DNA is 21% C and 21% G, I can subtract those from 100%, the total amount of the DNA, and I can figure out how much is left, 58%. Now, if we look at this 58%, how much is A and how much is T? Well, the key is we know that A and T always pair together. So half of this will be A. Oops, let's put an equal sign and a plus sign. So half of this will be A. And if A is 29%, then we know that T is 29%. Now notice we can always double check our math here, because if we add our, in this case, they told us C at the beginning, but we add C and A and G and T, this has to add up to 100%. If it doesn't add up to 100%, somewhere some of our math is off. And then finally, the third type of problem is something similar to what we were talking about, what we showed earlier. If we know one strand of the parental DNA, we can figure out the other strand, and then we can figure out what the two sister chromatids look like after replication. So these problems are your homework for next Tuesday. So I have to go to some meetings this afternoon, but let me know if you have any questions. And as I say, send me an email, or send me an email and we'll set up a time for a Zoom meeting. I can't meet this afternoon, but most of the other times this weekend I should be open. Are there any questions about how we would solve these problems right now? The other thing is, make sure that you're actually turning in assignments and turning them in on time. Some people aren't turning in assignments. Some people are turning them in half a day late. You get a little bit of leeway, but there is a 10% penalty for late assignments. So if you're consistently not turning something in or turning it in late, that's probably hurting your grade. Okay, if there are no questions, we'll stop sharing. I guess we should stop recording too.